Hello, everyone, and welcome to AMP one more time, AMP Part 2. In this session, we will cover the lymphatic system. So previously, what we've been talking about is basically the cardiovascular system, and it consists of the heart and the blood vessels, and the blood, obviously, and we understood that from the very beginning in the blood that we had the formed element, which consisted of white blood cells, red blood cells, and platelets. And then when we talked about the blood vessels, we talked about just touching what they did. The arteries carried oxygenated blood away from the heart, and the veins carried deoxygenated blood to the heart. The arteries, the aorta was the start of the systemic circuit, and then the veins, as it comes back into the aorta, were part of the pulmonary circuit. So we just kind of talked about the relationship between the blood vessels and the substances that go through there. And then we talked a little bit at the very end of that chapter in the blood vessels about the lymphatic system and how the lymphatic vessels are closer to the venule end of the capillary bed, and they take on excess plasma, and they have a role in the immune response. So this entire chapter is going to be devoted to the lymphatic system and this concept of immunity, or the body's ability to ward off diseases. So a couple of terms here before we get into any more of the anatomy or physiology. First term that we come across is pathogens. And you guys, if you're in a microbiology class or have had one recently, you're probably very familiar with this term. A pathogen is a microscopic organism that can cause disease. So viruses can be pathogens, bacteria can be pathogens, protists can be pathogens like amoebas, and you can even have multicellular animals called helminths or parasitic worms that can also be pathogenic. And it is the job of your lymphatic system to, in part, or one of the main jobs of it is to deal with those pathogens and to infer immunity. So what our lymphatic system does is that it protects us against disease, and that's the whole discussion of, conferring immunity. Lymphatic stem cells can respond to environmental pathogens. It can respond to toxins that are released from those pathogens. If you've taken a microbiology class or will soon be taking one, you're probably very familiar with toxins that can be released from different types of bacteria. From gram-negatives, they can have that lipid A toxin on their LPS layer. And your lymphatic system, surprisingly enough, will actually respond to abnormal cells. So your own cells that kind of get out of whack, and they may start to act as cancerous cells. So although your lymphatic system has is attuned to recognize when those cells just aren't behaving the way that they should, because cancerous cells grow so quickly and so rapidly, they outpace those cells in the lymphatic system that will fight these rogue cells. It's one of the reasons why your lymphatic system isn't able to completely rid your body of cancer alone, and that's why we have to outsource it with chemotherapy, surgery, and radiation. So how we organize the lymphatic system is that we have some very specific components, and we'll talk about the jobs of these components in more detail as we go through the chapter. So the lymph is a fluid. It's very similar in consistency to plasma. Big difference between the lymph and the plasma. One big difference is that the lymph is found in the lymphatic system, so that's why we call it lymph as opposed to plasma. And plasma is found as part of the non-formed elements in your blood. Also, another big difference is that plasma doesn't have any of those plasma proteins, those albumins, fibrinogens, immunoglobulins. It doesn't have any of those. I kind of hesitated on immunoglobulins because we will talk about B cells very shortly here and those antibodies. But they don't have those plasma proteins, those really big plasma proteins that we associate with the blood. Lymphatic vessels are the tubes or the vessels through which the lymph flows through. So it carries lymph from the peripheral tissues to the venous system. Remember, those lymphatic vessels, they kind of piggyback, or they kind of merge into the venule end of the circulatory system. We also have lymphoid tissues and lymphoid organs. So your tonsils would be considered lymphoid tissues. Your spleen is a lymphoid organ. And then you also have the cells of the lymphatic system. Lymphocytes, T cells and B cells, phagocytes, which are just like, they're monocytes that are now in the lymphatic system, and they can migrate in and out of circulation, so we just call them macrophages. And then we have other immune cells that we can find in here as well. So when we look at the class of lymphocytes here, and we're going to spend a great deal of time differentiating and talking about T cells and B cells here, what we notice is that those class of lymphocytes can divide, and in the blood chapter, we just talked about the fact that we had 5 classes of white blood cells. And those 5 classes of, or 5 different types of white blood cells fit into 2 classes of granulocytes and agranulocytes. And those agranulocytes were monocytes and they were lymphocytes. And that the lymphocytes had their very own stem cell that they arose from. Monocytes, eosinophils, basophils, red blood cells, and the other formed elements, even platelets, they came from myeloid stem cells, whereas lymphocytes came from lymphoid stem cells. And very briefly, we talked about how lymphocytes have more of a process of differentiation that we would talk about later on. So now we're going to talk about that differentiation. So lymphocytes can be divided into 2 classes. They can be T cells or B cells. So right here on this first slide, we're talking about those T cells. The T cells can then further differentiate into different classes of T cells. They can be cytotoxic T cells, which will attack foreign cells or body cells that are infected by a virus. So even if it's your own cell and it has a cell surface marker that says, hey, I'm a Kelly cell, but it's infected by a virus, so that cell surface marker is slightly altered, and we'll talk about that alteration in a moment. Cytotoxic T cells will target those cells for destruction. We have helper T cells. Helper T cells help to stimulate and activate both T cells and B cells, so just as their name suggests, they help, they are helpers. And then we have suppressor T cells, and suppressor T cells help to kind of monitor the activation of both B cells and T cells, and they make sure that we don't have that these numbers of cells don't get so large so quickly that they start to cause an autoimmune response or that they start to attack themselves. So it suppresses and inhibits the activation of T cells and B cells, and they kind of act as a regulator. So you may have heard suppressor T cells referred to as regulatory T cells because they kind of regulate and monitor these classes of lymphocytes, both B cells and T cells. And then we have memory T cells, and as their name suggests, and you can probably imagine what they do, is that they are a set of T cells that will respond to an infection that you previously encountered. With our next class, our B cells. So T cells confer what we call cellular mediated immunity. The cells will directly attack the problem or the pathogen or the infected cell or the abnormal cell. B cells are going to release antibodies. And another term that we can use for antibodies is immunoglobulins. So B cells differentiate into plasma cells, which will actively make antibodies. And then we have natural killer cells that are also another class of lymphocytes, and they're not as, what's the word I'm looking for, they're not as specialized as the other classes of T cells that we've had, or even the B cells. They are just kind of these circulating lymphocytes, and they're going to target basically any type of infection, so they don't really have that specificity that we'll see for T cells or B cells. So the lymphatic system and body defenses provide resistance to fight an infection, which we've already talked about. And we have 2 categories that we're going to talk about: innate or nonspecific defenses and adaptive or specific defenses. That's that specificity that we were talking about with B cells and T cells. Innate means natural or something that is instinctual. So innate immunity or innate defenses you're born with, they're going to fight any infection, any pathogen the same way. They're not going to be specialized for one particular type of infection. And adaptive defenses or specific defenses, these are immune cells that you've built memory for, or these are immune cells that have memory cells now that have been built up over time. And as you've lived your life, you've adapted to this environment so that the next time that you see this specific type of infection, you have the specific either T or B cells or both that can address this specific type of infection. So you've adapted to the environment and you have this adaptive defenses. Now, even though we're going to talk about these 2 categories of defense separately, keep in mind that it's not just that, we start with your innate defenses and then we exhaust all of your innate defenses, and then and only then will your adaptive defenses kick in and start to do their job. We kind of see there's going to be overlapping between both the innate and the adaptive defenses that are working pretty much simultaneously. So innate defenses, some key things to remember about them is that they're always going to work the same way every time, and doesn't matter what type of pathogen or invading agent that we have, and we consider that non-specific or innate resistance. Adaptive defenses, on the other hand, are going to be very specific, and they're going to protect against a very specific pathogen or infectious agent. It depends on the activities of the lymphocytes, so there's going to be a special class of B cells, a special type of antibody that is made in response to this defense. There are going to be special types of cytotoxic T cells that are adaptive or made in response to this particular type of pathogen. One way to think of it with your adaptive defenses, your body kind of says, fool me once, shame on you, fool me twice, shame on me. So it has these memory cells that are made so that they can respond to it appropriately the next time they see it. The understanding of our body's adaptive defenses is really the benchmark, or the kind of lays the groundwork, or is the precipice for why we have vaccinations. That, you know, you get a vaccination, you're exposed to just a little bit of this virus or this bacteria, and then your body makes memory cells in response to it. So let's look at some of your non-specific defenses. I think your book is going to talk about 7 or 8 of them. So starting from the outside in, we have physical barriers. Physical barriers are going to keep hazardous materials outside of the body. So your skin, lysozyme, which is an enzyme in your tears and in your saliva, the fact that you have your skin is very salty, and most bacteria don't like very salty places. All of those things are physical barriers that help to protect and keep hazardous materials from even getting inside the box or inside the body. Phagocytes are going to be these monocytes or neutrophils or eosinophils that have a phagocytic activity to them. They can remove dangerous microorganisms. You also have these natural killer cells that will participate in immunological surveillance, which means they kind of monitor your tissues, and they look for any cells that aren't displaying the appropriate, hey, I'm a Kelly cell marker. So there are specialized proteins that are in your cells that says, hey, that I'm supposed to be in this area. So these natural killer cells will ignore them, but if that marker is altered, changed, or missing, those natural killer cells will respond to those cells and attack them. We also have these chemical messengers, and we have interferons. So cytokines are specialized chemical messengers that will trigger the production of antiviral proteins in normal cells. And it doesn't matter what the virus is. It's not going to be specific for the virus. It's just for any virus-infected cell. Antiviral proteins don't necessarily kill the virus, but what they can do is that they can keep the virus from making more copies of itself or replicating inside the cell. So that helps to halt the proliferation of the viruses in your body. And then we also have complement. Complement is a cascade of circulating proteins that will, there's 3 outcomes that we have for complement. We can make the bacteria or virus sticky, so it's easier to phagocytize. We can also have, and that process is called opsonization. Complement can also trigger inflammation. So inflammation is kind of ringing the alarm and saying, hey, there's a problem over here. Let's get some more white blood cells in this area. Let's get some more immune cells in this area to fight this infection. And then we can also have what's called the formation of an MAC complex, or we can have these complement proteins literally poke holes into the bacteria and allow for osmotic lysis to take place. So there are several different things that we can have happen with complement, but they're always going to respond in the same way. And these are just circulating proteins that will respond to antigens, and antigens are cell surface markers on the outside of a cell. A foreign antigen will trigger either a specific or a non-specific response. And right now we're talking about the non-specific or the innate defenses, stuff that you're just born with, that you come here with. So if we have a bacteria, it's got its own specific cell surface markers, or we could also call them antigen, the fancy science name for it. Your body will notice these antigens are not supposed to be there, and it can trigger things like complement, it can trigger interferons, it can trigger natural killer cells to come into play to deal with this infection, and even phagocytes. Inflammatory response, we talked about that just a little bit when we talked about complement. That's one of the things that complement can do, is it can trigger an inflammatory response. So it is a localized tissue-level response that tends to limit the spread of injury or infection. So when you think of inflammation, you probably think of swelling, redness, tenderness to the touch for inflammation, kind of ringing the alarm and saying, hey, we need more of the immune cells to come to this area to fight this infection. In the meantime, we're going to restrict circulation so that this pathogen doesn't escape too far from this area. Also, we have fever. A fever is your body's way of trying to smoke out, quote unquote, the pathogens that are making you sick. It's a higher than normal body temperature. Most of the pathogens in your body like to hang around at about 37 degrees Celsius. And 37 degrees Celsius is your body temperature. So if your body temperature increases, it will cause denaturation of the proteins of these agents that are trying to make you sick, these pathogens. So if you denature the proteins, then you can kill the bacteria or keep it from engaging in its normal metabolic activities, which is going to kill the bacteria or kill what's trying to make you sick, bacteria namely. The drawback to a fever is that if it's denaturing the proteins that are in viruses or bacteria and inhibits them, then it's a good chance that fever is prolonged. If it's too high for too long, it's also going to denature your own protein. So it can be a double-edged sword there. A fever will also accelerate your defenses, and it increases your body's metabolism. All in the attempt to raise your body temperature to smoke out these pathogens that are making you sick. We have special types of histocytes or macrophages. We have microglia cells that we've talked about last semester that are found in the central nervous system. Culture cells that are found in your liver sinusoids, so those sinusoidal capillaries that we see these cells coming out of there, and they provide very non-specific defenses, and they'll fight every pathogen that they encounter the same way. We also have free macrophages, and free macrophages just kind of wander around and do their thing, and a special category of free macrophages are the alveolar macrophages, or what we call phagocytic dust cells, and these we find in your lung tissue, and we'll come back and talk about alveolar macrophages next chapter in respiratory again. So natural killer cells, remember, they are part of your nonspecific or innate defenses. They're defenses that you're born with. What they do is that they look to see the antigens on a cell, if they are normal or they're abnormal. If they're abnormal, then the natural killer cells can recognize and adhere to the abnormal cell. What will happen next is that the Golgi apparatus, which is going to release the proteins that have been made by the natural killer cell, and these proteins are primarily made to trigger either apoptosis or to trigger cell lysis in the abnormal cell. So those proteins that are made and they're shipped and packaged from the Golgi apparatus, they help to align with the cell so that they can directly impact the cell that's being targeted. So the specialized proteins, now we're going to call them perforin molecules, are going to form pores or a perforin complex in the abnormal cell in order to trigger osmotic lysis, or for the cell to have holes poked into it so it bursts. And then we have lysis of the abnormal cell. And remember, this is not going to happen. It's not that these natural killer cells are looking for a specific change in the antigen or the cell surface marker. They're just looking that the cell surface marker is either what it should be, hey, I'm a Jody cell, or what it shouldn't be, hey, I'm a Kelly cell. So if you have that's one of the reasons why if you have a tissue transplant, you're always going to have to be on some type of immunosuppressive drug to keep your immune cells from attacking this new tissue, whether it's a new cornea or it's a new kidney or whatever it is. You're going to always have to be on immunosuppressive drugs to keep your immune cells, things like your natural killer cells, from recognizing that the cells that are in your cornea or your kidney or wherever that you've received as a tissue donation are not going to be attacked and killed. So we've also talked about how interferons are part of your nonspecific defense. Interferons, when you think of interferons, I want you to think about ways to limit a viral infection. We have alpha, beta, and gamma. Things that I want you to remember about alpha interferons is that they're produced by leukocytes, or white blood cells, and they stimulate your natural killer cells, so they trigger them to be much more active and to proliferate more, or to have more division, so we can have more of them circulating. Beta interferons are secreted by fibrocytes, and the job of beta interferons is that they really help to kind of slow the inflammatory response, because as you've probably already experienced, that inflammation, just like a fever, has its role that we want it to play, but inflammation that's too widespread and it's too the duration of it is too long, is going to be really uncomfortable, and it's really not good for your regular homeostasis. So we have measures that are in place to slow down inflammation, and beta interferons are one of those measures we have in place. Gamma interferons secreted by T cells and natural killer cells are going to stimulate phagocytic activity. Our next nonspecific defense we'll talk about in detail is complement. And remember, complement are just those cascades of proteins, or a bunch of proteins that are activated in a cascade that can lead to opsonization, making the pathogen sticky or easier to phagocytize or to eat. Inflammation, which we've already talked about on the previous slides, the job of inflammation kind of ringing the alarm and localizing the infection. And then it can also lead to macular adhesion complexes, or the formation of pores into the bacteria that will cause for osmotic lysis to take place. Now, to activate or get this whole process started, because these proteins are always kind of floating around, but if you're not fighting off an infection, then you don't have to have those proteins activated. We want them to just kind of float around and just kind of wait until that they get activated. So in order to activate them, there are a couple of ways that we can do that. We have the classical pathway, and we have the alternate pathway. And there's another pathway that we can use that you'll talk more about that in microbiology. So the fastest method is the classical pathway. It's a fast method. The first complement protein, which we'll call the complement protein, instead of keep saying complement 1 protein, complement 2, we just give them a C. So C1 is going to actually bind to circulating antibodies that are already attached to an antigen. So let's say you have here, now remember before I talked about how your specific and your nonspecific defenses do not operate in a vacuum. Not like all the nonspecific defenses are exhausted, and then and only then do the specific defenses get to do their job. It doesn't work that way. So on this bacteria, so we're going to name him bacteria. If he is circulating in your body, he has cell surface markers that we call antigens. And as I said before, your specific and your nonspecific defenses are working in tandem at any point in time. So the antibodies, which are part of your specific defenses, they notice, and this is an antibody here, that there is a bacteria floating, and those antibodies are attracted to those antigens. And then this guy here is an antigen. So they notice that there is this antigen that's floating around, and these antibodies are going to be attached to it. Now, this C1 protein that's circulating around, it's then going to be attracted to the antibody that's attracted to this antigen, and we call this an antigen-antibody complex. So the C1 protein attaches to that, and that's going to begin this or catalyze this chain reaction of C1 being activated and C2 being activated and so forth. So here's a pretty picture of that classical pathway activation. Big things that I want you to remember about the classical pathway activation are that A, it starts off with the antibodies binding to the antigens in the bacterial cell wall. That's going to allow for the C1 protein to attach, and then it acts as an enzyme, and it activates the other complement proteins. And then the classical pathway ends when we convert the inactive C3. So the inactive C3 being converted to C3b that attaches to the cell wall of the bacteria is kind of where we're all going with this. Now for the alternate pathway, that C3b protein is just going to attach, it attaches to the cell wall itself. But for the classical pathway, and another thing that I want you to remember about this is that not only does it have to have the antigen-antibody complex, but it's the most rapid and effective activation of the complement system, the classical pathway. So this part is important, that part's important, and that once C1 is activated, catalyzes a series of reactions that activate other complement proteins, and that the pathway ends when we convert C3 to C3b. The alternate pathway is a little bit slower method of activating the complement. And when we say slow, this is relatively speaking. Keep that in mind, that it's not going to be like 45 minutes slow or anything like that. But it's slightly slower. And this is where the T, B, or D factors that are present on a bacteria will activate this pathway of complement. So the alternate pathway is important because some of these bacteria, parasites, or virus-infected cells are going to have these different factors on there. So it begins when many of these complement proteins are going to interact in the plasma, and then this interaction causes this interaction happened because there was this foreign substance in the body. So there were these bacteria that were present. And at this point, notice that for the alternative pathway activation, that we didn't have the antigen-antibody complex. We didn't have the antibodies recognize the pathogen first. So this is helpful because maybe we didn't have the particular antibodies. We've never seen this before. So if we've never seen it before, we wouldn't have the antibodies on deck to very quickly recognize it. And so we have to go through this clonal selection and clonal expansion to get the appropriate antibodies made, and that might take a little bit of time. So with the alternative pathway, the other way around this is that we have these infectious materials that are in the blood, and so these plasma proteins start to become activated, and they will attach to the cell wall. So we have the activations conversion of C3 to C3b, the same way we did at the end of complement, the classical pathway of complement activation. We still end with the C3b being attached to the bacterial cell wall. And as we just literally said 2 seconds ago, both the alternate pathway and the classical pathway are going to end with a conversion of the C3 protein to C3b. I'm going to star that or sunshine it because that's important. So for both of these pathways, that's going to happen. So those three effects of complement that we've talked about, we can form MAC complexes or macular adhesion or membrane attack complexes. 5 complement proteins join and they literally poke holes or form pores, and it causes the cell to go osmotic lysis to take place, more effective against gram-positives and gram-negatives. And if you've had micro, then you probably understand why, because gram-negatives have that outside LPS layer. Enhances phagocytosis by opsonization, so it makes the bacteria stickier. So these the complements working with the antibodies will kind of stud the bacteria, and it makes it easier for your phagocytotic cells to grab a hold of the bacteria and eat it. Some bacteria have capsules around them, which makes them slippery and hard to catch on to. So through this process of opsonization with these plasma proteins along with these antibodies, we can have this phagocytosis take place better. And it can also trigger inflammation or increase the degree of inflammation because histamine is released. The effects of inflammation are for one, which we've already talked about, it keeps the growth of the pathogens from going to surrounding tissue. So that's a swelling we have. We want to keep the pathogens from and localized. We don't want them to spread all over the place. It also rings the alarm. So it mobilizes the local and the systemic defenses so that we can have more red blood cells, or I'm sorry, more white blood cells come to the area, more immune cells, if you will, come to the area and deal with this infection. And then inflammation can also, because of the chemicals that are released, temporarily repair and develop and create a barrier against pathogens. So the redness, swelling, warmth, and pain, you are going to have that redness as a result of the dilation of the blood vessels to increase vessel permeability, to bring more immune cells to this area. Phagocyte attraction on there, so we are going to release cytokines or special chemical messengers that will, including neutrophils and macrophages, that will eat the pathogen, and at the same time those cytokines that are released can also activate your specific defenses, so those T cells and B cells can be activated again. And at the same time, because we are ringing the alarm that we're saying that, hey, we have a problem here, we're going to have fibroblasts come to the area and other cells to help regenerate and repair the tissue. So after inflammation, because there's been a lot that's going on there, there are going to be some byproducts. And one of those byproducts is that there's going to be some dead tissue there from the bacterial infection, and also from the fact that we've had immune cells that have come to this area, and any of those cells that were affected may have been targeted and killed, so we have some necrosis that takes place. So necrosis is one of the byproducts of inflammation. Pus, which is just kind of a mixture of cellular bacteria debris as well as necrotic tissue. And then an abscess is really just when pus accumulates underneath the skin in an enclosed space. In a perfect world, all pus should be pushed up and out and away from the body, but sometimes that pus is enclosed in an area and it has nowhere to go. It can't be pushed up and out and away from the body. When you think of an abscess, a lot of times you think of things like boils or an abscess tooth, because it's just not able to get up around the root or something. And then if an abscess is not treated, because there's no place for all that pus and stuff to go, then it can get systemic and you can have these things circulating in your body, which can be a problem. And you'll have to take some sort of broad-spectrum antibiotic, and sometimes even have the abscess drained. A lot of times actually have the abscess drained and take a broad-spectrum antibiotic to try to kill anything that may have gotten away. So fever specifically is pyrogens are any materials that will cause your temperature regulator, the hypothalamus, to increase the body temperature. Things that can act as pyrogens are circulating pathogens, toxins, and even antibody-antigen complexes, like the same thing that activated complement, the classical pathway of complement. We consider a fever to be anything where your body temperature is maintained above 37 degrees Celsius. We have endogenous pyrogen and interleukin-1. A pyrogen is released by active macrophages. So endogenous means that it's something that's in your body already. So the cells that are in your body, your active macrophages, will release this pyrogen, and it's a chemical messenger that tells your body to create a fever. So not only can circulating pathogens, toxins, or antibiotic complexes trigger a fever, we can also have interleukin-1 that's stimulated by your very own cells that says, hey, let's try to raise the body temperature in order to cook out some of these bacteria. So it's a chemical messenger, this endogenous pyrogen or interleukin-1, whichever you want to refer to it as. It's a chemical messenger that's sent to the hypothalamus, and the hypothalamus is kind of the control center for body temperature that says, hey, increase the temperature. But not only do we have this, but we have other things that can cause a fever as well. And anything that causes the hypothalamus to raise the body temperature, we call it a pyrogen. So now that we've talked about all of the non-specific defenses and we've talked about exactly how they work, the physiology of how they work, and we talked about the anatomy of the cells that were responsible, now we're going to talk about specific defenses. A good short answer question would be to compare and contrast 4 of the non-specific defenses. What do they do? What is their physiological role? How do they operate? How do they work? What is the anatomy of them? What kinds of cells are they coming from? Are there macrophages, fibroblasts? What are the chemical messengers that are specifically released? And what is the effect on the body? So those sorts of things, you know, comparing and contrasting 3 or 4 different types of non-specific defenses would be a good short answer question. So now on to the specific defenses. So specific or adaptive defenses, as the name just says, are going to respond to a specific antigen. This is going to be the coordinated effort or action of your T cells and your B cells. Both T cells and B cells came from the same kind of white blood cell that we called a lymphocyte. So there are a couple of different forms of immunity that you probably already kind of understood just going over the non-specific defenses. The first type is that you have innate, which is present at birth. We've talked about that. Then you have adaptive, which are, we kind of called it your specific immunity here, and this is just after you've been born and you're living in the environment, you get exposed to all kinds of pathogens and all kind of crap, and then your body infers some memory to it, and you've adapted to this environment so that the next time you see this pathogen, you're equipped with the memory cells to fight it and probably won't even get sick. We also have this concept of active and passive immunity. Active immunity can either be active immunity can be either where the antibodies develop after exposure to the antigen, so it's under the umbrella of adaptive immunity. So after you've seen the chickenpox, and then the next time you're around someone with chickenpox, you don't get it because you have memory cells against it. Or we can have this idea of passive immunity. Now, we're still talking about antibodies. Like antibodies, you develop your own antibodies after you're exposed to the antigen. But in passive immunity, this is where we just take someone else's antibodies. And in passive immunity, we can do that. What's happening there is that someone else has made antibodies, and then you get those antibodies injected into you so that you confer some sort of immunity. We also see that with pregnancy from mother to child. Mother gives some of her antibodies from all the things that she's seen. They can cross the placenta, and they can confer some immunity to their unborn child. And also in breastfeeding, that's kind of what we consider a natural passive immunity, known as baby didn't have to make baby's own antibodies against this pathogen. Baby was able to get antibodies directly from mom via her breast milk or via the placenta. In either case, their antibodies are transferred, whereas if you get the infection or you get a vaccine and you actively make your own antibodies, we consider that active immunity. So for active immunity, it can be naturally acquired or artificially induced. Naturally acquired means you were hanging out with your buddy, he had chickenpox, you got chickenpox, so just through environmental exposure. Artificially induced active immunity, you still made your own antibodies. That's the whole key here for active immunity. You make your own antibodies. Artificially induced active immunity means that you went to the doctor, you were scheduled for a vaccination against chickenpox, so you have through vaccine that contains the pathogens, then you made your antibodies from it. So you can either get it in the wild, naturally acquired, or get it in the doctor's office, artificially induced. For passive immunity, naturally acquired means we get it from the mother via the placenta or via the breast milk. And then artificially induced just means that maybe you have rabies or you contracted rabies. And one of the treatments for that is to get the antibodies from either a horse or rabbit serum, and they inject those antibodies into you. Four properties of immunity. One we've already talked about, specificity. for specific immunity, because we're under the specific immunity umbrella now. So specificity, versatility, memory, and tolerance. So those are 4 properties of your specific immunity. Versatility is important because we're going to see various different types of pathogens and bacteria, and if you're taking a microbiology class, you're very well aware of the fact that there are lots of differences between even the same genus of bacteria. So these immune cells need to be very versatile. We need to have some that will stick around and remember this for next time. Remember, fool me once, shame on you. Fool me twice, shame on me. And there needs to be some tolerance that your body doesn't adversely respond to itself, so that it's able to recognize cell surface tissue. Now, for some people, they may have an autoimmune disease, such as lupus, the autoimmune disease, rheumatoid arthritis is an autoimmune disease, where their bodies, some of their immune cells, are not recognizing specific types of tissue and will attack it themselves. I even read an article somewhere, it has been a few years ago, where it was kind of questioning, there was a research question whether or not the overuse of certain types of medication, was it triggering more autoimmune diseases? Was it triggering more people to have autoimmune diseases or not be able to have the same type of tolerance? So first we're going to talk about T cells. T cells, and remember for T cells, and I'm going to go back and show you that slide real quick just to jog your memory about T cells here. We have the cytotoxic T cells, helper T cells, suppressor T cells, and memory T cells. T cells are part of what we consider your cellular mediated immunity. So in your textbook, in your notes, we'll talk about cellular mediated immunity. Cellular mediated immunity is going to be where the cells themselves are actually targeting the cells that are causing a problem, if you will. T cells only recognize antigens that are bound to glycoproteins in the plasma membranes. These glycoproteins that are in the plasma membranes are cell surface receptors, specialized cell surface receptors that we call major histocompatibility complexes. Whew. Long word, long phrase there. So we're just going to shorten it as MHC. So these MHC markers, we have 2 classes of them, MHC1 and MHC2. They are found on the plasma membrane of all of your cells, or mostly all of your cells. And they are genetically encoded on chromosome number 6. So since they're genetically encoded, these special proteins, these glycoproteins, are going to be differentiated among individuals so that you don't have the same MHC1 or 2 markers as your child, even though you gave birth to that child. You don't have the same as your mother. You don't have the same as anyone. They're genetically unique to you, this major histocompatibility complex. T cells will only recognize antigens when they're bound onto these MHC complexes. So unlike what we saw for complement proteins and antibodies, we saw that they can recognize these different antigens all on their own. T cells don't have that same ability to recognize antigens that are just floating around, or cell surface markers on cells that aren't supposed to be there. They have to bind to an MHC protein, or a major histocompatibility complex protein. We have 2 classes of them, MHC1 and MHC2. MHC1 class proteins are only going to be found on your cells that are nucleated. So any cell in your body that has a nucleus is going to have an MHC1 class protein, or MHC class 1 protein. Think about some cells that we've discussed in this semester already that don't have a nucleus. They lose their nucleus as part of their maturation process. Red blood cells, right. So we don't really find a MHC class 1 complex on your red blood cells. Class 2 are found in antigen-presenting cells. So these are specialized cells, are usually macrophages, that they can take an antigen in, or a substance that's not supposed to be in your body that we'd find in a bacteria or something, or a virus, these macrophages can take an antigen inside, have it bind with its lysosome, which has a bunch of chemicals in it to digest it, break that antigen up, and then take a piece of that antigen and display it on its class 2 marker, and it shows those T cells, like, look, this is what we're dealing with. This is our problem in sector G. This is our problem. So now that the antigen is bound to an MHC marker, the T cells can recognize it. So, and we also find these class 2 markers on lymphocytes as well. So 2 types of MHC markers. Class 1 found in all nucleated cells. Class 2 is only going to be found on specific types of cells, those antigen-presenting cells and other lymphocytes. Class 1 MHC proteins are going to pick up small peptides or protein fragments that were found inside the cell that shouldn't be there, like fragments of viruses that are trying to replicate inside the cell, or fragments of bacteria that are replicating inside the cell, or that have somehow come in contact with the inside of the cell. These class 1 MHC proteins will bind to those antigens, and then they will display it on the surface of their cell. And what they're displaying, what they're saying to the immune cells is that, hey, I'm a sick cell. I'm infected with a virus or a bacteria or some sort of pathogen, and this is what it looks like. So now, because we have the antigen connected to an MHC1 protein, the T cells are going to be able to respond to it. Normally, if that MHC1 protein is just displayed on the surface of the cell and it doesn't have any antigen attached to it, then the T cells are going to ignore it. It's going to ignore those normal peptides, those normal proteins. It's not going to really think anything of it. There's nothing attached to it. But once an antigen is attached to it, then it will trigger the T cells to destroy that cell. Even if it is your own cell, it destroys that cell because the signal the cell was sending was that, hey, I'm sick, destroy me. So here's a pretty picture of that. We have a virus or a bacteria that is attempting to replicate on the inside of the cell. It's going to make its own little viral or bacterial, so it could be a virus or a bacteria, either one. It's going to make its own little abnormal peptides. When we make our MHC1 marker, which is the blue thing here, it's going to bind to some of those abnormal peptides, and when it's time to be displayed on the cell surface of the cell, it's sending a message that says, hey, I have an MHC1 marker that says I'm supposed to be in this body. However, I also have an antigen attached to it. So when an MHC1 protein has an antigen attached to it, then that says the cell is saying, hey, I'm sick, kill me. So a little video here, and I'm not sure how well this audio is going to work. If the audio doesn't work very well for you, let's see what I can do here. I don't even think I can play it. Let's see. Won't be able to play this video, but it just shows that whole process of antigen antigen processing for a cell that has an antigen presented on its MHC1 marker. So in your notes, make sure that you, because it's in your notes that are online, make sure you view this video. So MHC2 proteins, remember that this class of proteins is only found on lymphocytes and antigen-presenting cells. So what happens here is that the phagocytic cell, whether it was a class of lymphocytes or it was a macrophage, is now going to bring that pathogen in, break it down, and then present it on the surface of the cell. So instead of saying, hey, kill me, when we have an antigen that's on a class 2 MHC marker, it's telling the T cells to kill this. This is the antigen that is causing us problems. So if an antigen is on an MHC1 marker, the cell is saying kill me. If an antigen is on a class 2 MHC marker, it's saying kill this. So here we have that happening. So the phagocyte, the phagocytotic cell has phagocytized this bacteria. It merges with its lysosome, kills that bacteria, breaks it down. Some of the antigens are going to bind with this MHC class II protein. When it binds with that MHC class II protein, what it does is it says, hey, T cells, this is our problem. Kill this. This is the issue we have. Big difference between class 1 and class 2 is that when you had an antigen presenting a class 1 marker, that bacteria or virus is already replicating inside the cell. The cell, notice, didn't have this binding with the lysosome. So there was no lysosomal action take place. The cell that was infected was infected. It didn't phagocytize the cell. It didn't try to kill it. So it was an infected cell that needed to die. In this case here, notice we have a lysosomal action taking place. We're phagocytizing it. We're actively eating it and breaking it down so that there is no bacteria any longer. There's no virus any longer. We just took a part of it and displayed it on our MHC class II marker to present the antigen, if you will. It's why it's called an antigen-presenting cell, to say, hey, this is our problem. This is what we need to kill. And once again, I don't know if you guys can ever see these YouTube videos that I post when I put them in the Collaborate lectures, so go ahead and make sure that you view those as well. So we're going to stop there for today, and then we will talk more about what happens after the T cells recognize the antigen on a class 1 or a class 2 MHC protein. Have a great day, and we will finish this up in another session.